Hi there. In this video, we're going to see how cells make glycogen. So glycogen or glycogen is the sugar forming, the one that gives rise to sugar compound. And it was identified by Claude Bernard some time ago. Now, how does it look like? Each one of these molecules here is a glucose monomer. And we have, and the glucose monosaccharides, they join each other in alpha 1,4 glycosidic bonds. So this is the 1 carbon, this is the 4 carbon, and we have the 1,4 glycosidic bond. Okay, and the anomeric oxygen, of course, is in the the anomeric carbon is in the alpha anomer configuration. But in addition, and then you have a chain of those. But in addition to those 1,4 glycosidic bonds, you have 1,6 glycosidic bonds, which are also alpha. So you have the alpha carbon here. This is the oxygen here, the hydroxycarbon at the 1 position, forming a 1,6 bond. That's the 6 carbon, okay. Carbon 6 and glucose. So you have chains and branches coming off that chain. Okay. Where is it made? It's made in the liver, a lot of it, and provides glucose in the circulation for many other other cell types and organs. There is a lot in muscle, but muscle doesn't share it with anyone. Okay, it doesn't get so glucose from muscle doesn't get glucose from the glycogen in muscle doesn't get into the circulation. There are small amounts also in other in other cells and tissues. So why store glucose into this kind of polymer? Why don't we just have them as individual glucose, single molecules of glucose, as opposed to this polymer of glucose? That's what glycogen is. It's a polymer of glucose. So the reason is because it's a good mobilizable reserve for glucose, as you will see. We'll explain why that is. And when you really want to put something in storage, you want to have tight packing. Okay, every time you've started putting stuff in some storage space, you really can't just throw stuff around on the floor. Okay, you really have to pack it very tightly to make good use of the space that you have. And as you do so for molecules such as glucose, you want to do it in a way that you do not cause osmotic stress in the cell. So glycogen fits both of these criteria. It does provide for tight packing of glucose molecules, and it does not cause any osmotic stress. So how does glucose get put into glycogen? Okay, how does it get stored into glycogen? And if you look at the glycogen, it will look like this. You have all these chains and the branches coming off, and all of them are nucleated, as it turns out, by a protein. And that was not discovered until maybe 20, 30 years ago, which is very surprising, given how much we knew and how much glycogen had been studied for centuries, pretty much. The fact that there is an actual protein at the core of every glycogen globule, if you wish, was quite something. So that protein is called glycogen. Okay. So what does it do? We'll see what it does in a few steps, but let's start with what happens when you want to put glucose into glycogen. So again, the whole pathway begins when you don't want to take glucose into glycolysis or any other use, like in the pentose phosphate pathway, but you want to put it away. So you had a big meal, there was a ton of glucose into your circulation, you take it because it's good stuff, good food, you don't just let it go through your digestive system, and then you put it away. So the first thing you do is to phosphorylate it with the hexokinase or glucokinase at the 6th position. But while this is good, because it's a substrate for all the glycolytic enzymes, as we've seen 1,000 times by now, that's not good enough for glucose storage. You have to take that phosphate and move it to the 1 position with phosphoglucose mutase. And you can see where this is going. I mean, because before you can do anything with glucose, you have to activate it in this case. And we've seen already how the activation works, and we're going to see it again. But anyway, to activate glucose, you need to have it to have the phosphate on the 1 position. So the enzyme that does it, we've already seen it, so we're not going to go into too much detail. We've seen it. We saw it in galactose utilization, going in that case from right to left, from glucose 1 phosphate to glucose 6 phosphate. here we're going to go, as I said at the time, this is a completely reversible reaction. So we're going to go from glucose 6-phosphate to glucose 1-phosphate. And the way it is happening to change the phosphorylations here is to have an intermediate that it's doubly phosphorylated. So the enzyme contributes one of these phosphorylations. So you have an intermediate that's doubly phosphorylated. And then the phosphate that was there before at the 6th position is been moved onto the enzyme. So what you're left off with is the phosphate that was put on by the enzyme at the 1 position. And now you got glucose 1-phosphate. Again, nothing new here. We've seen this before. And now, why we did this is now you need to activate glucose before you can do anything in terms of glucose of glycogen synthesis. And we've already seen this reaction. I mean, how do you activate it? You have to uridylylate it. In this case, you have to activate the sugar. And again, what happens here? You have glucose 1-phosphate. The phosphate here, one of the oxygens on the phosphate on glucose 1-phosphate will attack the alpha phosphate of UTP. It can be any trinucleotide, really. Okay, but in this particular reaction, what we need is a UTP, and so the two terminal phosphates, the beta and the gamma, leave as pyrophosphate, and then they get hydrolyzed. So you have it drives. This reaction is used to drive unfavorable reactions. So you have a lot of delta G, negative delta G getting released. And what you end up with is 1-phosphate from glucose 1-phosphate plus the alpha phosphate that was left. And now you have really UDP. You have 2 phosphates here. And of course, the nucleoside part of U being attached to glucose. So you end up with the UDP glucose and pyrophosphate. And now that activated glucose, you can use it to add it onto other things. And in this case, you add it to glycogen. Okay. So glycogen is a protein, as I said, and it gets glycosylated in this case. So glycogen and glucose, that's how it's going to be modified with glucose. How does it get modified? Well, glycogen is actually the enzyme itself. So it modifies itself, okay? It is a glycosyl transferase. We've already seen something like this with galactosyl transferases when we're making, let's say, lactose, in mammals. So, and I told you then that there are many glycosylatic enzymes, in that case, galactosyl transferases that put galactose onto various substrates, various proteins. Here we have a glycosyl transferase that puts glucose molecules on proteins. So it's a glycosyl transferase itself, and it's a dimer, and it is in the core of glycogen. And glucose is added at tyrosine 194. So it's a dimer, so there are going to be 2 tyrosines that are going to be glycosylated on glycogen. And in more detail, this looks like this. This blue part here is glycogen. That's the protein part. And there is a tyrosine, which is a hydroxyl group. And it is that hydroxyl group that will attack and the UDP glucose. Okay. So what you're going to get is glucose here through the 1 carbon position here being attached to tyrosine, and you release UDP. Okay. So UDP is released. Here, there it is. It goes away. And you have glycosylated with 1 molecule of glucose, glycogen. You keep doing this. You do this 3 more times, and you extend glucose by extra residues, extra monomers of glucose. So you end up with 4 in the end. On this tyrosine, you have a short chain of 4 glucose molecules. Right? Every time, again, UDP glucose comes in. So you have another UDP glucose coming in, another one, and another one, and you have 4 on glycogen. On 1 tyrosine glycogen, it's a dimer, so you do this, twice. And then you end up with glycogen that's glycosylated. Now the big gorilla comes in, which does kind of the similar reaction that glycogen did, but it does it much more efficiently. That's glycogen synthase, and also it takes UDP glucose. I mean, no matter what you do, to put it into glycogen, glucose has to be uridylated. Okay, and so and glycogen synthase will take it and extend it. Okay, adds more and more and more, more than 10. So it's a very efficient enzyme. The question is, why bother with glycogenin? Why couldn't glycogen synthase come in and start just extending? I mean, why do we need glycogen at the first place? I mean, to have this short little stump here of 4 glucose residues. Well, the answer is that glycogen synthase needs a landing pad before it can work. So it can extend a pre-existing short chain of 4 at a minimum glucose residues, and does it very efficiently, as I said. But it still needs this extra little primer, of which it can extend. So this is why we need glycogen, okay, to provide, and that's why it was such a big discovery when it was discovered, because it was known that glycogen synthase has this requirement of a minimum of 4 pre-existing glucose residues in a chain before it can extend them further. All right, so that's the idea here. That's why we need glycogen, and then glycogen synthase comes in. But let's assume, okay, let's think about it for a minute. I just told you that you have glycogen as a dimer, and you have 2 little stumps of 4 glucose residues extended from it, and then glycogen synthase comes in and extends. If that's all glycogen synthase could do, or I mean, that's all it can do. But if that's all there was to making glycogen, what you would end up with is glycogen at the core, and then 2 giant strings of glucose chains extending. That's clear, not a very tight packing. You have 2 super long, chains just swiveling around in the breeze there in the cell. So that's not good. So you need to do something else. So far we have seen glycosyl transferase, as I said, which they transfer activated carbohydrate, moieties in this case, sugars, and from donors to acceptors. And now the activated means usually they're nucleotide related. In this case, it's UDP glucose. We've seen it start, it's ADP glucose. Okay, it doesn't matter. The acceptors could be anything. We already saw a protein one, which was glycogenin. And in this case, we have a pre-existing glucose synthase, a glucose chain. How does it work? So we'll answer the question in a minute. What else needs to happen to make glycogen, the tightly packed glycogen globule? But before we get to it, this is how the enzyme works: glycogen synthase. And in principle, if that's your activated glucose molecule, that will be added to some acceptor. The acceptor can come in from different ways, based on from different sides of the closed form of the sugar molecule that's uridylated in this case. Depending on which side it comes in, it comes from, the orientation. Now, you're not going to retain the anomeric form of what you had, or invert it. So the top is an inversion. So it comes from the other side, and you start here with the oxygen here pointing down, and all of a sudden at the end, you have the oxygen pointing up, right? So depending on the mechanism, and some of them do that, okay, some glycosyl transferase, but you can retain also the configuration if it comes this way, shown here from the same side. And this is possible, and then you have the same configuration here. Look at the anomeric oxygen here going down on the anomeric carbon, in both cases, when you start with and what you finish with. The telltale sign of which one is happening is this intermediate. If you have a retaining mechanism, what you need to have is an oxygen bound to three things at the same time. That's an oxonium ion, and it's an intermediate, and that's really, if you detect that, then you have a retaining, and that's how we know that glycogen synthase follows a retaining mechanism. So the configuration of the anomeric carbon one does not change with glycogen synthase when it makes glycogen. All right, and so that's the idea. So you keep adding, all these residues, and that's the intermediate. So this is the activated glucose that comes in for every extension that you do, for every single one you add. And the UDP leaves, it generates oxonium intermediate, which is attacked by the 4 carbon oxygen on the chain that was already there on glycogen. But all right, so how do we solve now the 2 giant strings flopping around the wind? Well, we have branches, much like a tree that has many little branches of, let's say, even a few major ones, and then you have the full canopy. So what we really need to make here is a canopy again. It's like the tree analogy is actually very apt. It generates a full-blown canopy of, if you wish, a glycogen globule. So how do we branch? So the branching enzyme comes in, and for the branching enzyme to work, you already have to have at least 11 residues. So glycogen synthase has to have worked already, extending the four, let's say, glucose chain that the four residues of glucose that were on glycogen, it extends it to a minimum of 11, and then and only then the branching enzyme comes on. It clips 6 or 7 of them and moves them to carbon 6 of another glucose on the chain. Why 6 or 7? Because that will leave at least 4 back, which means that glycosynthase can go back and extend whatever was trimmed, right? So first, before you can make a branch, you have to trim. So you have a longer chain. There is a core glycogen, that's its 4 residues. Glycogen synthase comes in and extends it. And now the branching enzyme comes on. Okay, we have 1, 2, 3, 4, 5, 6, 7, plus 4, 11. That's the minimum. Okay, that needs to happen before the branching enzyme comes on. And then the branching enzyme trims and clips 7, 6 or 7 from the end. So it leaves at least 4 behind, right? And then takes the 6 or 7 and puts them on the 6 carbon. Okay? So you have a 1,6 glycosidic bond. That leaves now 2 ends from which, guess who? Glycogen synthase can come on and start extending both. And once it extends them even more, you have at least 11, then the branching enzyme comes again, and the whole thing repeats again and again and again, and then that's how you form a big round glycogen globule. Why do we do that? Of course, you pack better, okay? You increase the solubility, there are more ends for glycogen synthase to work, and most importantly, okay, there are more ends from which you can mobilize glucose when you start trimming your glycogen globule, and you want to do this as fast as possible because, let's say, you need glucose to run away from a predator or chase a prey or something like this. So you really need to mobilize as fast as possible your reserves. So the more ends you have for your enzymes to work, the fastest this mobilization will be. And you keep doing this, as I said, this whole process, and with the branching enzyme, cycles of branching enzyme and glycogen synthase keeps happening again and again, and you end up with a properly formed glycogen molecule. Okay? So it's a pretty cool way. In the next video, we're going to see how now you use that as a source of glucose. So how do you degrade it? So, but we'll stop here at this point.